I'm trying to get these pieces. What is this?
Oh, we actually like took a break from drawing. Yeah, for CC. Oh, yes, that was interesting.
Can we take
So that was really cool. Yeah. Yeah, it was. We got to meet like. Oh my god! It's like it's so good. No, I was like four. Was there?
Not looking too bright.
I should record and send. Oh, you didn't.
Oh, right.
I know. Yeah. So because they don't. I think they don't want us to.
They want us to.
They do want us to. I know. I thought that a lot.
Everyone able to complete the quiz?
Yeah.
So, we will finish up the rest of the slides now, and then we'll have time to do the problem setting. So the procedure each lecture period. So we're talking about different chemical interactions right now. Some of these are probably going to be somewhat familiar, some may be new, but we need to understand these types of bonding, mostly non-covalent type interactions. Those are the interactions that hold together the structures of large mass molecules and also allow for interactions between molecules. And they're non-permanent. So that you can have a receptor and a ligand coming together and then apart and so on. So there tend to be low energy interactions as if they're more fluid. Looking here at hydrogen bonding. Hydrogen bonding is the interaction between a hydrogen and an acceptor molecule that has a slight negative charge on it. So that's sort of what this designation is. This hydrogen has a slight positive charge on it, and this acceptor has a slight negative charge on it. This is the hydrogen bond. The reason why that interaction is occurring is because of these slight positive and negative interactions. And the reason why those are in place is based on what these atoms are bound to here. In that this is a covalent bond. This hydrogen bound to its oxygen is a covalent bond. Those are sharing electrons, but they're sharing them in an unequal way. And so that is called a polar covalent bond. And so you have, because of that, oxygen is what's called an electronegative atom. It tends to pull the electrons more closer to its atomic center, its nuclei. And so it has a slight negative charge to it, and therefore it leaves the hydrogen with a slight positive charge. So this non-polar covalent bond creates the scenario where this hydrogen has a slight positive charge, and therefore it can be a donor to a hydrogen bond with something else, which is also in a polar covalent bond. In this case, this carbon that is double bonded to the oxygen is a polar covalent bond as well. The electronegative atom here has some unpaired electrons. It has a slight negative charge to it. This is the hydrogen bonding here. Okay, so the hydrogen bonding is not in here or here, it's between these two. And it's a very weak bond. But if you have lots of them throughout a structure of a molecule, like we see here, this is it looks a lot like a DNA helix, but it's actually a helix of proteins. And in this case, these green dots here are hydrogen bonds. And they're basically between atoms that look like this. A little bit different in that this is a nitrogen bound to an oxygen, and this is a carbon bound to a hydrogen. This is a, yes, carbon bound to an oxygen and a nitrogen bound to a hydrogen. Those are in polar covalent bonds of their own, here and here. And so it creates that scenario where you can get a hydrogen bond interaction. If you have lots of those throughout a structure, it can really help stabilize the whole three-dimensional structure of the molecule. So they are important, especially at scale. Okay, so these are the types. This is not something you need to memorize, but these are the types of molecules that can participate in hydrogen bonds with each other. Things like water. So one sort of side of water, an oxygen and a hydrogen, can be the donor to another side of water, which is the oxygen itself and covalently bound to a hydrogen. So there's hydrogen bonding that can happen between water. And that, we'll talk about that in a minute, that creates really specific characteristics for chemistry of water, in that it has lots of bonding between it. But these are other types of molecules. On this side are the donors. It is always a hydrogen, which is covalently and polar, covalently bound to an electronegative atom like oxygen or nitrogen. And then the acceptor tends to be something that is electronegative itself, either having some unpaired electrons or just electronegative on its own. So those are things you will see as common donors and acceptors. And then the hydrogen bonding itself are just these dots between them. The solid lines are covalent bonds. Questions on that concept, Rick? Okay, so looking at water and how water as a molecule can be both a hydrogen bond donor and an acceptor. This is the structure of water. So we have oxygen, which is bound to two hydrogens, as you know, but then it also has two unpaired electrons here. So oxygen has a slight negative charge to it. It's electronegative, and it also has these unpaired electrons. So there's a slight negative charge to it overall. And the bonds here covalent, but they are polar, in that the oxygen tends to pull the electrons more closely to its own nucleus. That leaves these hydrogens as having a slight positive charge. So this end of the water, would this be a donor or acceptor here? That would be a donor, right? The positive charge end here, this would be the area sort of donating to it, a hydrogen bond, and then you have potentially two acceptor locations sort of on this end. And so you can get these lattices of hydrogen bonding between water looking like that. Okay, so each water molecule can basically create 4 hydrogen bonds. Okay, so those aspects sort of act like a glue for water. There's lots of hydrogen bonding between them, and therefore, if you want to change the state of water, you essentially have to break those hydrogen bonds. So you have to input a lot of energy in order to do that. So that's why if you're looking at a comparison here between other atoms that are bound to hydrogen, like methane, things like this, their melting point and their boiling point is much, much lower than water. That's because this compound doesn't have any hydrogen bonds between it and other things, whereas water does. So water's many, many hydrogen bonds make it require a lot of energy in order to melt and then also boil. Okay, so the temperatures are much higher. Okay, so water has these interesting characteristics that are facilitated by all these hydrogen bonds, but the polar nature of water, meaning having a slight positive and negative charge to it, also has implications in the body. it itself is a polar solvent, meaning it likes to associate with other polar molecules, either things that have positive or negative charges too. So it likes to dissolve things that have and associate with things that are positive and negatively charged. And we sort of know this intrinsically. We know that water hides away from things like lipids, right? They form globules in the water, they don't dissolve, and things like salt do. Okay, and so what's going on when you mix salt, which is a crystal here, of sodium ions and chloride ions, which are bound together in ionic bonds, salt dissolves in water because it can interact with either of those ions on its own. So if we mix salt and water, the chloride ions, which have a slight negative charge, which end of the water molecule do you think they'd be associating? Right, the hydrogen, right? The hydrogen has the slight positive charge to it, so it is interacting well with the chloride. And what about the sodiums? Which end of water would those be interacting?
Oxygen.
The oxygen, exactly. So you can dissolve your salt into water to a certain extent until it's saturated and every single water is interacting with the salt, and then the salt falls out of solution. It's no longer interacting with water. Okay, so those are charged molecules like salt that is interacting with water, but you can also have just general polar molecules, something like this, where there isn't a true positive or a true negative charge to them, but there is a polarity. So any molecules that have polar, meaning slight positive or slight negative charges, can also interact well with water. They're called hydrophilic. They like interacting with water. Okay. And so we will sort of think about that, especially when we get into amino acids that are polar or charged. Those are those that like to interact with water, and amino acids that have no charge, they're neutral, don't want to interact with water. Okay. So we'll get into this in a little more application type details. Sure. Okay, so thinking about hydrophobic interactions. Hydrophobic molecules are those that don't interact well with water, and typically that's because they don't have any charge to them. Water likes to interact with charges, as we just talked about, and so if a molecule doesn't have any charge, meaning its bonds are just covalent, there are no slight positives or negatives, those molecules tend to hide from water. Okay, and so what happens, we can think of that as like mixing oil and water. The oil tends to form globules, right, inside water. What's actually happening is this. So you can imagine, and this is sort of related to energetics and things liking to be disordered. So there's sort of a level of entropy that things like to go towards. And in this case, water likes to have entropy. It likes to be able to move around and change position and not be locked into a certain position. What happens if you have nonpolar molecules, those that don't have interactions with water, is that the water essentially is turning away from them. It's not able to interact with them, and it forms cages like this. Now, these are water molecules. This is a hydrophobic molecule inside. The water is not interacting with that molecule. It's doing everything it can not to interact. It's only interacting with its neighbors. But because it doesn't interact well with this, it can't fall out of position. It's sort of locked into this cage because it's trying really hard not to expose any of its charged surfaces to this hydrophobic molecule. So it's locked into this cage structure. And if you think about the drive towards randomness and random movement, these cages are not a very favorable situation. Okay, so the hydrophobic effect is to basically, when you're mixing a hydrophobic molecule with water, is to limit the number of cages. So that limit the number of times water has to be ordered like this. And so what happens is that nonpolar molecules tend to glom together so that the cage structure is limited in size. Instead of having two, like here, two nonpolar molecules, two ordered cages of water, they tend to come together. They're not really bonding together. They're just kind of in the same area. And the number of waters that are ordered around them is small. Okay, so that's called the hydrophobic effect. Hydrophobic molecules tend to come together to limit the ordered structure of water. Questions on that? It's called hydrophobic effect. It's not called hydrophobic bonding or anything like that because there aren't any true bonds between these molecules. Okay, so then we've sort of talked about hydrophobic molecules and hydrophilic molecules, those that interact well with water and those that don't. We have also structures or molecules that have both sets of characteristics, and those are called amphiphilic. So they have some region associated with them that is hydrophilic and another region that is hydrophobic. And we talked about these briefly last time, but these are, they can be amphipathic lipids. They have a group on them, phospholipids we talked about a little bit, where there is a group attached to a lipid. This line here is just a line of carbons, and then they would have hydrogens attached to them. So very nonpolar ends here, hydrophobic. There's no way for those to interact well with water. The head groups, though, what do you notice about those? They're charged, right? So they have some charge to them in different ways. They have different atoms and so on, but we know that those charges are going to interact well with water. So the head groups there are hydrophilic, the tails are hydrophobic, and we see these in phospholipids that are in membranes. So a cellular membrane would be composed of a lipid bilayer, and those are the tails sort of hiding away, and then the heads can interact with water. And that's good because outside the cell is aqueous and inside the cell is aqueous. So you have what's called a lipid bilayer. You have two sets of tails facing each other, and the heads interact with the heads can interact with water on both surfaces. Okay, and we'll look at the structure of a membrane. So this is what this looks like. And so if you mix a lot of molecules like this, if you had some sort of tube of lipids like this, and you put it in water, they will self-assemble into membranes. Things like this, like this. And what they're doing is that the blue part here is the polar head group, the part with the charge on it, and it will interact well with water. And then these tails, which just have carbons and hydrogens, no charges, are hiding. Okay, so they will assemble into things like vesicles, and this is a bilayer. You have two layers of the lipids. They can also form a monolayer at the surface where the tails are just pointing up and the heads are just resting on the surface of the water. So you can actually see these form on their own. And that's the hydrophobic effect, essentially, is what's going on. People can use this process to develop nanoparticles, things that they're trying to deliver drugs to cells with. You can package up, let's say, an aqueous drug inside the internal component of this vesicle, and then you can deliver it into the bloodstream, and it can fuse with cellular membrane. Use case of that. Okay. Questions about amphipathic molecules or membranes or anything? We'll get into membrane structure as a whole, but right now just looking at amphipathic type molecules. Okay, and then not to alarm you too much here, but a quick review of chemistry in terms of acids and bases. this is going to be important because we are going to be looking at especially amino acids that can be acidic or basic. We want to understand the nature of acids and bases and how they react in a chemical way. So acids are proton H plus donors. They can lose a proton in solution. And strong acids, this is an acid, a generic acid with its hydrogen. In that form, it's called associated. It's associated with its proton. And then it can go back and forth and become dissociated. In this case, the acid is dissociated. We have its so-called conjugate base. This is its base form. It's lost its proton, and this is the free proton. Okay. So every acid has a tendency to dissociate at certain pH, and that tells you how strong an acid is. Its acid dissociation constant. So it's saying at equilibrium, what is its tendency to be in this form, dissociated, losing its proton, versus the concentration in its associated form. So stronger acids will be those that have a higher concentration of just free protons at equilibrium. Okay. And there is, you can look at charts of acids and their acid dissociation constant, how likely those acids are to come apart. And that will tell you how strong it is. So if you're looking at this chart, what is the so-called strongest acid? The acid with the largest Ka value tends to dissociate more frequently. Yeah.
Phosphoric.
Yeah, so it looks like phosphoric acid here has the largest Ka value. And you can relate this directly to its pKa. So what do you think a pKa is relative to a Ka? How would you relate those two? About hydrogen and pH. How do you get that? What's the calculation you do this? Yeah.
Negative log.
Negative log, exactly. So it's negative log of the Ka gives you the pKa. And what the pKa tells you, this is actually a number that, if you think about a little more intuitively, it is the pH at which the acid is half in its associated form and half of it has lost its proton. So that tells you that at pH 2, phosphoric acid, half of it has its proton, it's associated, and half of it has lost its proton. That's really acidic. So it means that even at pH 2, half of it is losing its proton. So that's a very strong acid. These other ones, like, let's see, dihydrogen phosphate, those are closer to pH 7. So those are more sort of half dissociated physiological pH, about pH 7. So we will also think about these pKa's in terms of amino acids, because some amino acids can gain and lose protons, and that's important for their chemistry too. So in terms of catalyzing reactions and things like that. So everyone understand what Ka means and what pKa means. We tend to talk about pKa more, so just keep that in mind. So we're thinking about pH and pKa's and dissociation and things like that. We want to think about, most of the time, we're talking about living systems, which are about a pH of 7.2 to 7.4, except in certain parts of the body or in certain parts of cells where we have gastric acid, stomach acid is about pH 2 or a little lower, very acidic. And then lysosomes, so organelles in the cell that digest proteins, can be more acidic as well. The general blood chemistry is around 7.2 to 7.4. So controlling the pH of our body is very important. We do not want to become too basic or too acidic. And we'll get into why that is important. It can affect our proteins and it can affect our lipids and things like that. When you start peeling protons off of amino acids, it can change the structure of protein. So there are lots of other reasons as well, but keeping our blood chemistry around neutral is very important. Okay, so thinking about a pKa and association and dissociation. So we just defined that at a pKa value for any one particular acid, it is half in its associated form, meaning half of it has its proton bound to it, and half of it has lost its proton. So that is at a pKa that's specific to a particular acid. So this molecule, this generic acid, has a pKa of 4.2. So at 4.2, half of it is associated and half of it is dissociated. All right, so if we have that acid and we start to put it in a more basic solution, we are raising the pH. What do you think is happening to the 50-50 split between the associated and dissociated? If you're adding base, what do you think is going on? As the pH increases, the base that you've added is basically pulling protons off of the 50% that had its proton still, right? So what happens is as the pH increases more and more and more of it becomes dissociated because the base that you're adding is pulling that proton off. Questions about that concept? Now, conversely, if we decrease the pH, meaning when you decrease the pH, you're basically adding free protons to the solution, right? You're adding an acid itself. If you're adding acid, what happens is that the part that was in its base form, it's going to accept protons from that. So the 50% here that was dissociated is going to say, okay, I'll take the protons back, and more of it becomes associated. Okay, so there's going to be a change in the percentage that is has a negative charge to it or is associated, depending on the fluctuations of pH. We will get into this more, especially when we talk about amino acids and their dissociation state. So, but any questions on that before I move on? I want that to be okay, so let's look at this in the case of this glob, which is a protein. Okay, so we have a big string of amino acids that this protein is made up of. And like I said, amino acids can have different characteristics. They can be nonpolar, they can be polar, they can be acidic or basic. So an acidic or a basic amino acid is one that would donate a proton or take a proton, right? So you can imagine if you have an acidic or a basic amino acid, it is going to have a pKa itself. Okay, and there's only a few of them, but there are a few that can donate or gain protons back. And so you can imagine that their protonation state is going to change depending on what the pH is the protein is sitting in. Okay? So if you have a protein that is at essentially physiological pH, the isoelectric point of this particular protein is 6.8. That is the pH at which overall that protein is neutral. It has some negative charges and it has some positive charges, but overall it's neutral. Okay. So some of its groups have a positive charge, some have a negative charge. That's neutral. Now this protein, if you then put it in a more basic solution, removing the pH that the protein is sitting in, what's going on to the amino acids that can lose or gain protein? Adding base here, what do you think is going on to those side chains that can donate protein? So if you're adding base, yeah, whatever acidic groups are in the protein are going to donate it to the base. That the solution is in. So this solution that's becoming pH 11, if there are any amino acids here that can donate protons, they're donating it to the base. Those protons are being pulled off. Okay, and so overall, this protein becomes negatively charged. The more basic the solution, the more negatively charged the protein is because it's donating protons, which have positive charges away. Okay. Now the opposite would be the case if you move this protein into a more acidic solution. What do you think is going on here? Why is it becoming more positively charged if it's in a more acidic solution? Yeah, any groups that are negatively charged are pulling the protons from the acid. So they are being proton acceptors. And if they're proton acceptors, they're gaining positive charge overall. Okay. So conceptually, start to wrap your head around that. So what can happen though is that when that happens, the characteristics of that protein really change. The way it's folded up. If it relied on positive, negative charges to fold up, that's going to fall apart. If it relied on positive charges here to interact with other proteins, those are gone if they're in a basic solution. So the characteristics of the protein change wildly if you put it in different pHs. And that can be really important to its biological function, right? If your body gets up to pH 11, all of your proteins are negatively charged. They're probably all at full. That's not great. So anyway, we'll get into that, especially when we start talking about amino acids, but wrap your head around the concept of proton donors and acceptors and how those change their state depending on the pH. Okay, questions on that before we do a problem set? That's about as much chemistry as we can. Remind yourself of those topics. Okay, so let's go into the